Well, we've managed to look at the various characteristics of dementias and then took an in-depth, focused look at the two most common forms of dementia, namely vascular dementia and Alzheimer's disease. We saw that Alzheimer's disease is by far the most often diagnosed form of dementia. And therefore, it behooves us to take a look at what the cause of Alzheimer's disease might be. And the fact of the matter is, we don't know what the cause is. And we have, over the course of the last 50 years or so, generated a number of theories that have come and gone in our attempt to find the cause of this very, very devastating disorder. So what we're going to look at now is sort of a history of the theories of Alzheimer's disease. We're going to look at a number of the most important theories, and I have them arranged on the slide in front of you in roughly chronological order, going from the oldest theories to the newest, if you will, current theories of what Alzheimer's disease is all about. And you can see they involve, there's 6 main theories we're going to look at. So let's get started at exploring the nature of each of these theories and seeing what their current status may be at this time. So the oldest theory we're going to talk about is the aluminum toxicity hypothesis. And as the name suggests, the theory purports that Alzheimer's disease is related to the toxic effects of being exposed to aluminum. So where did such an idea come from? Well, it began with a few epidemiological studies that tried to, from a correlational approach, understand why certain dementia-like disorders were occurring in a few Pacific islands and the Indonesian island chain. And, you know, they looked at as many environmental variables that they could at the time. And one of the variables that showed up to have a strong correlation between communities that showed this dementia-like disease and communities did not had to do with the concentration of aluminum in their drinking water. And this idea got into the literature. Some people examined the brains of Alzheimer's diagnosed victims and indeed found that their brains did seem to have concentrations of aluminum ions embedded within the senile plaques that are, if you'll recall, is one of the signatures of the Alzheimer's disease diagnosis. And perhaps the most important study that took this idea and moved it forward was a somewhat infamous study that was published in 1965 that revealed that when you take rabbits and you expose them to high levels of aluminum, that they in turn develop toxic neurofibrillary tangles. So there was sort of the smoking gun that showed an experimental manipulation that linked neurofibrillary tangles to, if you will, aluminum ions. This caused a lot of notoriety at the time. There were lots of stories in the press about using aluminum cookware. Soda pop cans that used to be steel were just beginning to be transitioned into aluminum, and there's lots of concern about that. And so there's a bit of hysteria in the community. And companies like Alcoa actually began to donate some money for Alzheimer's research. And in subsequent years, it was quickly determined that there did not appear to be a causal relationship between aluminum and dementia. And that whole idea of aluminum toxicity has been pretty much put to bed by the major scientific community. However, there are some scientists who continue to study the relationship between the accumulation of metals in the brain and dementia-type illnesses. And the ions that they're focusing on are iron and copper. but nothing definitive has developed in those particular lines of research. So in a nutshell, we'll say the aluminum toxicity hypothesis has essentially sundowned out and really is no longer influential in the way we think about possible mechanisms of Alzheimer's disease today. And now as we advance chronologically, we come up with a new idea that was somewhat revolutionary at its time. And it is an important idea, and it's the idea that perhaps Alzheimer's disease is a prion disease. So let's begin with the idea that virtually all infectious agents that we know of, bacteria, viruses, they proliferate, they reproduce using nucleic acid mechanisms of biology, namely either DNA or RNA. And it turns out that there are several forms of dementia-like diseases in both animals and humans, namely scrapie in the sheep, bovine spongiform encephalopathy or mad cow disease, and its human equivalent, Creutzfeldt-Jakob disease. These are rare diseases that can be transmissible, that you can actually in the laboratory transmit them from one animal to another animal. And the mechanism for this transfer is not based on DNA or RNA. Back in 1982, Stanley Prusiner discovered a new form of pathogen that had not been described before. And he called this new form of pathogen a prion. And he coined that term, and prion is just short for a proteinaceous infectious particle. That is a little piece of if you will, protein that can essentially lead to an infectious disease. And by the way, this work was so novel and so significant, Prusiner was awarded the 1997 Nobel Prize in Medicine and Physiology for this work. So since that discovery, many researchers hypothesized and studied the possible role of prions in Alzheimer's disease. If it can cause dementia like Creutzfeldt-Jakob disease, maybe Alzheimer's disease is some sort of prion disease. So let's try to wrap our minds a little bit around the whole notion of a prion disease. The first thing we need to know is that prion proteins are found in all humans. They're an important part of the constituents of our brain. However, when the prion protein, the base prion protein, mutates into a rare conformation, that is, normally it's found in certain shapes, but for reasons we don't fully understand, occasionally it somehow mutates into an abnormal shape. When it mutates into this abnormal shape, it begins to exert some new emergent properties that we didn't see before in the protein. That misfolded protein causes other related proteins to become misfolded as well. The process slowly spreads from molecule to molecule, and that's what a prion disease is all about. This spreading wave of misfolding of proteins. And as you may recall from your basic biology, the way most biochemical processes in the body occur, in enzymatic processes, they occur due to locking key, if you will, fits between adjacent molecules. And if the molecules don't have that lock and key fit, they can no longer interact and carry out complex chain reactions in biology. So all these misfolded proteins can no longer sort of participate in the function that they evolved to mediate. And that causes a breakdown in the system and a disease. Well, as people began to explore the notion that prions could be involved in Alzheimer's disease, they quickly began to become discouraged that this was a useful way of conceptualizing the disorder. First of all, it was observed that the rate of progression of the spread of the misfolding, if you will, that one saw in, let's say, Scrapie or mad cow disease, or, if you will, Creutzfeldt-Jakob disease, the rate of the spread in those disorders is much faster than one saw in Alzheimer's disease. So something very fundamentally different seemed to be going on in these two different classes of disorders. Also, there was no evidence whatsoever of being able to transmit Alzheimer's disease from essentially one human to another, or from one mouse to another in mouse models. And then finally, in the 1980s, when the senile plaques, which are a signature of Alzheimer's disease, were finally figured out what proteins they were composed of, namely the beta amyloid protein, it was found that these proteins were much, much smaller in molecular size than any of the known prion species. So all those facts together seem to suggest that a prion approach was not the way to proceed in trying to understand the basic nature of Alzheimer's disease. And as a result, funding for this approach, this hypothesis, kind of dried up. And the whole idea of the notion of Alzheimer's disease as a prion process was pretty much forgotten by the next generation of scientists who came along who were studying these types of issues. But as we'll see in the very end of today's lecture, there's a bit of an update here, and that is this sort of forgotten idea about Alzheimer's being a prion disease is beginning to get a new life, because there have been some great disappointments in the major contemporary theory of Alzheimer's disease, number one, but also some recent discoveries that perhaps the beta amyloid proteins, the tau proteins that have been identified as important players in Alzheimer's disease, they may actually have subspecies forms that resemble the behavior of prions. So there's kind of like a new interest in looking at the potential role of prions in mediating the disease that we call Alzheimer's. And we'll get back to this, I think, in the very last slide of this presentation. Almost in a contemporary, if you will, parallel time of the consideration of the prion hypothesis, the cholinergic hypothesis emerged. In the late 1970s, a renowned Alzheimer's researcher named Peter Davies and others demonstrated that the brains of Alzheimer's disease patients suffered from a marked loss of synapses, that is, connections between adjacent neurons that were mediated by the neurotransmitter we call acetylcholine. It was demonstrated, for example, that Alzheimer's brains had a 90% reduction in acetylcholine transferase, and that is a molecule that's a precursor to, if you will, the building or the synthesis of acetylcholine, and also a 50% reduction in the quantity of acetylcholine itself. So in Alzheimer's brains, they universally show a remarkable reduction in acetylcholine precursors and acetylcholine itself. And in addition, one of the earliest casualties in the brains of Alzheimer's disease victims that we see is a destruction of a subcortical area of the brain called the nucleus basalis. And it turns out that the nucleus basalis is a major site for the production of acetylcholine in the human brain. So all these things have come together to essentially suggest that a major sort of part of the Alzheimer's disease syndrome is this systematic, if you will, devastation of the acetylcholine system. And it turns out that the cholinergic mechanism, this systematic destruction of cholinergic neurons, has been the focus of much research. And although it's proved to be a good descriptive model of what happens in Alzheimer's disease, it is not a good causative explanation. That is, it tells us what happens, but it doesn't tell us why it happens, and that makes it somewhat limited in its scope. But nonetheless, I point out in this slide, the cholinergic hypothesis has been successful in terms of generating the development of pharmacological interventions, that is, medications for the treatment of the cognitive symptoms of Alzheimer's disease. So let's sort of explore how this whole intervention process works. We begin with the idea that acetylcholine is a neurotransmitter that when it's secreted from one neuron, it helps stimulate nearby neurons. So it's used by one neuron to communicate with another neuron. That's the basis of information processing in the brain. And when the acetylcholine is secreted by the sending neuron into the synaptic cleft, the space between it and the receiving neuron, it is quickly decomposed by an enzyme called acetylcholine esterase. So the transmitting neuron secretes acetylcholine, and that acetylcholine will stimulate the next neuron in the chain, but it only has so much time to do that because acetylcholine esterase will come along and destroy it, gobble it up. And that needs to do that in normal, if you will, everyday functioning, so that the time span of any acetylcholine, if you will, release is limited, as opposed to being persistent for a very long time, and therefore it won't be able to carry, if you will, lots of information. So it turns out that 4 of the 5 FDA-approved medications for the treatment of cognitive symptoms in Alzheimer's disease have been developed on the basis of the cholinergic theory. Namely, 4 of these 5 medications, the way they work is they inhibit acetylcholine esterase. And the idea is if you inhibit acetylcholine esterase, the little bit of acetylcholine that still remains in the Alzheimer's brain will essentially have a longer time period to sort of do its thing, to be able to stimulate the next neuron in the chain. And essentially those 4 drugs that use essentially the acetylcholine esterase inhibition strategy are Aricept, Exelon, Razidine, and Nemzaric, which we saw earlier when we talked about Alzheimer's disease symptoms and characteristics. So again, the cholinergic hypothesis has been pretty successful in terms of generating some interventions. However, what we have to remember is that these improvements that result from these drugs are really, really very small, and they're very, very temporary in terms of the overall scheme of the cognitive decline that's associated with Alzheimer's disease. And also, it's somewhat limited, that is, the cholinergic hypothesis is somewhat limited by the realization that the great majority of neurons in the brain are mediated by other neurotransmitters than acetylcholine. For example, glutamate and GABA, as we saw earlier in the first unit of this class on aging. So in a nutshell, we can now say, although the hypothesis was instrumental in generating some, if you will, medications that have a mild improvement in Alzheimer's symptoms, it really no longer plays a crucial role in the generation of new research, and it's sort of been supplanted by other theories. And we'll take a look at the major theory that's replaced it in just a moment. But before we need to do that, we need to look at another theory based on a neurotransmitter, and that is the glutamate hypothesis. And the glutamate hypothesis is sort of a mirror image of the, if you will, the cholinergic hypothesis. So let's begin by sort of wrapping our mind about what glutamate is. It's a major neurotransmitter in the human brain. And when a neuron secretes glutamate, it will interact with receptor sites found on nearby neurons. And in particular, the receptor sites that it stimulate are what we call the NMDA receptors. That's short for N-methyl D-aspartate if you're interested, but everybody just says NMDA because it takes too long to write that long, if you will, organic chemical term. So glutamate stimulates NMDA receptors, and when those NMDA receptors are activated, that causes a cascade of activity in the receiving neuron that causes calcium ion channels to open up. And what happens is the neuron absorbs those calcium ions, that depolarizes the neuron, which in turn triggers it to start firing to, if you will, stimulate other neurons in the chain. And what the glutamate hypothesis proposes is that in the Alzheimer's brain, there is an excessive output of glutamate. There's whatever is controlling glutamate, if you will, release is somehow disturbed. And that excessive release of glutamate causes the nearby neurons in the chain to essentially suffer what we would call calcium ion toxicity. They absorb so much calcium that it begins to poison the neuron. So the glutamate hypothesis has generated research which has led to development of 2 FDA-approved medications for the treatment of Alzheimer's disease, namely Namenda and Namzaric, which also, if you will, has a acetylcholine esterase inhibitor as well. And these drugs function by binding to the NMDA receptor sites and preventing their neurons from being activated by glutamate. That is, they're NMDA agonist. And if you tie up the NMDA receptors with a molecule that doesn't quite stimulate them but blocks glutamate from forming a lock and key fit, what happens is you prevent the calcium ion toxicity effect. And like the cholinergic class of medications we described earlier, these NMDA agonists provide only very mild and only temporary mitigation of cognitive symptoms. And so like the, if you will, the cholinergic hypothesis, the glutamate hypothesis, primarily a descriptive model of what happens and remains quite limited in its ability to generate additional research, if you will. And that brings us to our penultimate theory that we're going to be considering, namely the genetic mutation hypothesis. And this idea is pretty straightforward. There's, first of all, several lines of evidence that have come together to suggest that Alzheimer's disease might be the result of some sort of inherited genetic mutation. And what are some of the lines of research that sort of started people thinking along those lines? Well, there are several family clusters around the world that have been studied that have a highly inheritable form of early onset Alzheimer's disease. And so that suggests a genetic mechanism. And also it's well known that persons with Down syndrome develop a form of dementia resembling Alzheimer's disease sometime between their 40th and 50th birthday. And so together, both of these lines of evidence suggest that it might be fruitful to look for a genetic mutation that could be mediating these forms of Alzheimer's-like dementia. And the interesting thing about this little cluster of evidence is that it kind of gives investigators a head start of knowing where to look. And that's because Down syndrome is due to problems on chromosome 21. So when investigators at the National Institute of Health began to try to find this genetic explanation for these early onset forms of Alzheimer's disease, it was not too long before they found that on chromosome 21, the gene for the amyloid precursor protein, which is now known to be involved in the formation of the senile plaques and the neurofibrillary tangles that are the signature of Alzheimer's disease. And where did they find it? Right where they began the search, on chromosome 21. So this was somewhat of an interesting sort of jumpstart to this search for a genetic mutation. However, it quickly became quite obvious that this mutation that was seen in these early onset forms of Alzheimer's disease could not be found among individuals suffering from the most common form of Alzheimer's disease, namely the late onset variant that becomes more and more likely as we grow older and older and older beyond age 65. And the only other gene that's been identified to date that's associated with any increased risk of Alzheimer's disease is a particular variant of the APOE gene that's found on chromosome 19. And although one can find a weak statistical relationship with that E4 variant of that gene, there are many people who, in fact, the majority of people who inherit that gene never go on to develop Alzheimer's disease. So there's essentially no gene or gene cluster that has been essentially identified to date that can essentially predict who is going to get Alzheimer's disease to any particular degree of strength. People are still examining the genome in a number of different ways, but right now there is no quote unquote gene for the late onset variant of Alzheimer's disease that we've been talking about most of this time. Now this brings us to the big kahuna in the pantheon of Alzheimer's disease theories. And that's the beta amyloid protein hypothesis, or the abnormal protein hypothesis of Alzheimer's disease. And the amyloid precursor protein, APP, is found in all human brains. And remember, that was the protein that was found on chromosome 21 we talked about earlier. And the function of the amyloid precursor protein is not fully understood, but it is concentrated in the synaptic connections between neurons, and it appears to be involved in the formation of synapses, of new synapses, and the maintenance of old ones. And as I just mentioned, the gene for this, interestingly, is found on chromosome 21, the same place where we saw the mutation explaining early onset Alzheimer's in Down syndrome and in those family clusters that have been studied around the world. So this makes if you will, the amyloid precursor protein a good target as a possible mediator for the, if you will, onset of Alzheimer's disease. And so it's been studied quite extensively. So let's take a little closer look at the amyloid precursor protein and how it might be involved in this, if you will, theoretical mechanism of Alzheimer's disease. The first thing we need to know is that APP is constantly being produced in our brains, and that when it's kind of used up, the old APP is cleared out. And the way it's cleared out is it's kind of broken up into pieces, and then there are mechanisms that essentially enrobe those pieces and clear it out of the brain. It's sort of a brain waste removal system. And this is where things begin to go wrong in the Alzheimer's brain, in this breaking down and removal of, if you will, the waste APP components. So let's sort of try to follow along with the logic of the hypothesis, starting with step one here in this slide. The first thing is that there's an excess of an enzyme called BACE. And BAC enzyme stands for the beta amyloid cleaving enzyme. And what BACE does is it takes this long, if you will, amyloid precursor protein, and it breaks it apart into two components, a beta amyloid component and a gamma amyloid component. And this is not the normal way that the APP is broken down. When it's broken down by BACE, that amyloid component that results clumps together into the so-called senile plaques that we see as the signature for Alzheimer's disease. And those clumped together, if you will, agglomerations of beta amyloid cannot be cleared from the brain. And with this accumulation of beta amyloid, we begin to see toxic effects on the neuron. And this includes the breakdown of tau proteins inside the neurons themselves, which in turn aggregate together to form neurofibrillary tangles, which is the other signature of Alzheimer's disease. So we see in this beta amyloid cascade occurring inside of the brain, if you will, the genesis of both the senile plaques, now known as amyloid plaques, and the neurofibrillary tangles, now known as tau tangles, that are the quintessential diagnostic signatures of the dementia we call Alzheimer's disease. So this idea of this beta amyloid toxicity has been the dominant theory that's been guiding Alzheimer's disease research for the last two decades, particularly research whose aim is to develop a medication to treat the cognitive symptoms of Alzheimer's disease. And the strategies of this treatment research has primarily been of one of two varieties. Either a strategy that tries to prevent the formation of new beta amyloid accumulation, or essentially strategies to try to reverse the existing accumulations of these beta amyloid clusters. And it is not if you will, an overstatement to say that literally scores and scores of clinical trials have been performed on many, many, many varieties of drugs that have been developed to either prevent beta amyloid accumulation or to reverse beta amyloid accumulation. And billions of dollars of investment have been put into these clinical studies, and they've all failed to yield essentially what we could call a successful treatment. None of these have essentially research attempts have produced a drug that's been approved by the Federal Drug Administration to have significant enough improvements of Alzheimer's, if you will, disease symptoms to essentially meet a cost-benefit criterion. And this has resulted in great disappointment in the research community. Part of the disappointment is because we were really, really excited, let's say 15 years ago, that we were on the cusp of developing a treatment for Alzheimer's disease because of some of the robust predictions and findings in our preclinical models based on this beta amyloid protein hypothesis. But we've reached the point now with all these disappointing results that we have to ask ourselves the question is the beta amyloid hypothesis dead? Has it reached the end of its usefulness? And towards that end, let me read for you a quite profound quote that appeared in a recent editorial in the New England Journal of Medicine, one of the leading medical journals in the world. And this is a quote from Paul Murphy, who's a PhD researcher. And essentially Murphy says, quote, "It would be foolish to ignore the continued failures of the anti-amyloid approaches," that is to research. "In spite of a mountain of evidence supporting the primacy of the beta amyloid in Alzheimer's disease, researchers are coming to the realization that our preclinical models of the disease might be missing the mark. There's something that we really don't understand about what's going on here." So we look at a summary of some of the contrary evidence, against continued, if you will, pursuit of the beta amyloid protein hypothesis. It includes the dismal performance of the clinical trials. And indeed, it has been dismal, despite the best minds working on these problems and billions of dollars of investments. There's also the interesting finding that the mouse models that have been developed, that is genetically altered mice whose brains accumulate massive amounts of beta amyloid, so we can use them to study potential drugs for the treatment of Alzheimer's disease, that our mouse models that produce these excessive beta amyloid accumulations in the brains of these rodents are not accompanied by cognitive deficits in those mice. The mice with profound amounts of beta amyloid in their brains can learn a maze just as quickly as a control mouse. So there's a bit of a disconnect there. And relatedly, many postmortem studies that have been done on older people who have died of causes other than dementia have found that they might have massive amounts of amyloid plaque accumulations in their brains, yet there was never any evidence of memory or cognitive deficits in them before they died. So there's certain disconnects going on here that clearly suggest that we're missing some important piece of the puzzle in terms of the beta amyloid explanation of the genesis of Alzheimer's disease. And this brings us to our last slide in our consideration of the theories of Alzheimer's disease. And that is that there's been some recent evidence that suggests that if we combine the basic ideas of the beta amyloid hypothesis together with that old prion disease hypothesis that we considered earlier in today's lecture, if we combine those two together, we might be able to cobble together a, if you will, a constructive explanation of why the clinical trials conducted so far have failed. It's because several investigators have shown that both the beta amyloid protein and the tau proteins associated with Alzheimer's disease appear to have subspecies of protein complexes that behave like the prions that we saw earlier in Creutzfeldt-Jakob disease. That is, there seems to be subspecies of beta amyloid protein and tau protein that act like contagious proteins, prions, if you will, that cause misfolding of proteins in and around themselves in the brain. And that spread occurs without the need for DNA or RNA. So by reconceptualizing Alzheimer's disease and the beta amyloid tau protein processes that occur in that disorder of Alzheimer's disease, and thinking of it as a dual prion disorder, that is, there is a beta amyloid prion and a tau protein prion that might be causing the Alzheimer's disease, this helps explain why the targets of the medications used in the clinical trials that have been conducted so far have failed, and also generates lots of new ideas and new strategies for developing new generations of drugs that might be effective in treating Alzheimer's disease. So we went from our great high of expectations in about 2005, 2010, to today being somewhat disheartened in terms of when we can expect an important drug to come along that can significantly reduce the symptoms of Alzheimer's disease. So maybe this dual prion idea is going to help to get us to where we need to go. And so I would just suggest to you, in the meantime, we need to keep our fingers crossed and hope that the great expenditures of resources and the great numbers of very, very smart people who are working on the problem of Alzheimer's disease, that they will have some success in the not too distant future. I'm confident that they will eventually be able to treat Alzheimer's disease. What I'm not so sure about is the time frame in which that's going to unfold.